So my name is Lita Proctor. I'm with the National Human Genome Research Institute at NIH, and I'm the HMP coordinator. And I just want to make a couple comments. I really appreciate, Dushko, you're organizing this international congress. And I don't know if you did this, but I did. I did a head count of all the invited speakers, and of, I think, roughly 41, 16 of those, so a very large fraction were HMP. So that was terrific representation of HMP. And then secondly, I also did a second head count. We talked with Dushko earlier during the organization of this Congress to see if we could bring in as many next generation scientists in the HMP area as possible. And I did a second head count. Of those 16 HMP invited speakers, I believe 10 of them fell into that assistant professor postdoc category. So I'm grateful to the HMP investigators for sending their junior scientists from their laboratories, and I appreciate Dushko allowing the next generation to be presented at this international meeting. Thank you. So you've heard about this. I'm not going to go into terrific detail, but I just want to point out a very important thing about the HMP. It's always meant to be a community resource project. So the Common Fund, which is the kind of a familiar term for the office in the NIH Director's Office that funds this activity, is really, these activities are really meant to be catalytic. They're not meant to be long-term sustaining support for the programs. So this is was always meant to be a catalytic activity, and in that regard, the interest was to develop community resources of a wide variety in order to provide this kind of information, tools, and data for the community. So for example, we have a wide variety of repositories of not only sequence data, but also strains, clinical phenotype data, nucleic acid extracts, and so on. There are 6 different initiatives which are conceptually kind of described here in this inner circle. I'll get to in a minute. And all those initiatives within the HMP interacted within a very large consortium of 200 plus members. And that was actually another opportunity to bring in people who weren't necessarily part of, let's say, a major genome center to come in and help us make sense and analyze the data sets that we were producing as a community resource. So I'd have to say that's another major benefit of the HMP, was really helping grow the human microbiome community in the US. We also were a founding member of the International Human Microbiome Consortium, and of course you can learn quite a bit about the HMP on the STAC website. I'm not going to go into detail, but I want to point out that another unique feature of the HMP is the fact that we actually examined more than one body site simultaneously. We investigated 5 major body sites, and the goal was to characterize the populations, the microbial populations associated with these 5 major body sites. And that was done in sort of 2 ways. One was with the so-called healthy cohort study, in which every body site was clinically verified to be free of overt disease prior to sampling, which is a very hard standard indeed to set for a consortium, for a cohort. 300 adults and so on, and you heard quite a few talks these past 3 days about this study. We also had another set of human studies that we fondly call the demonstration projects, because we wanted them to be a demonstration of microbial associations with particular diseases. In this case, they fell into sort of 3 classes of major body sites: skin, GI tract, and urogenital, and you also have heard some talks here and at other International Congress meetings about these projects. So we're coming to the end of HMP, but please keep in mind that our goal has always been to provide tools, resources, repositories, data, and scientific approaches for the community. So there's been a lot of rumor about a possible HMP2. Just like the other programs, we're not at liberty to discuss any details, but of course the community has been engaged in giving us advice, the NIH advice, about what, if there were an HMP2, what are the needed resources, what are the limitations in the field that the NIH could be in a position to help develop. So I'm going to continue to talk about this as a community resources project. So if there were an HMP2, I think we're still visualizing it as a community resources project. I think that's a fair assessment. These are the kinds of ideas that have come up in the last year, not so different than what Patrick talked about, of course, because the community recognizes the needs. There's no mystery to what's needed in the field. So we held over the last year both in a series of internal conversations as well as a series of open meetings, and the Common Fund also had a social media open comment period in order to solicit comments about an HMP2. So without necessarily being able to tell you specifically, but there would certainly, if there were an HMP2, there would be certainly a very strong desire to develop the bioinformatics infrastructure as well as the computational tools, and I'm sort of separating those two because we're very interested in supporting system level analysis and, of course, data standards and appropriate data resources. Very general, but I can tell you that much. Secondly, we're quite interested in continuing to develop and promote high throughput, large scale assays for analyzing the microbiome. Very likely the next phase would be to develop functional property assays. And not only in the microbiome, but of course in HMP, we sort of ignored the host side because we're very interested in concentrating on analyzing the microbiome side of the story. So we're considering including more of an effort to analyze the host side of the story. Model systems, which was not a feature of HMP1. And also, the thing we talked about over and over again this past 3 days is the need for broadly consented cohort studies, prospective cohort studies. And I'm just going to stop there. Thank you.